All right, so we are going to start this lecture today with disorders of the immune system. And there are four major categories of immune mechanisms. The first is what we would call humoral or antibody-mediated immunity, and this is controlled by B lymphocytes. Then you have cell-mediated immunity, and that has to do with T lymphocytes, the complement system, and then phagocytosis, which is going to be your neutrophils and macrophage, which are also referred to as phagocytes. And we're going to go through each of these mechanisms as we go along. So the types of immune defenses are basically one, innate or non-specific immunity, and so you can just call these your natural resistance. This is what you were born with. And then the adapted or specific immunity a lot of times is referred to as your second line of defense, and this is more of what we might consider our actual immune system, and it's much more effective than our innate immunity. So talking about innate immunity, some of the components of this would be, for instance, epithelial barriers, which are your skin, your mucous membranes. They're helping to keep the bugs out. Imagine if you didn't have these, how easy it would be for you to have infections. And then, of course, phagocytes, and these are like little Pac-Men that are running around in the system, and they're eating things up. And so, like, for instance, you have phagocytes in your lungs, which their whole job is to move around through the lungs and eat up dust cells. And so a lot of times these phagocytes are just referred to as dust cells. And then you have opsonins and cytokines. Now, opsonins are chemicals, and some of the opsonins would be like antibodies, and these chemicals can bind to certain bad guys that get into the system, bacteria and viruses, dust cells, things like that. And then what it does is it makes it easier for phagocytes to grab onto these things and then by phagocytosis to eat them up. Then you also have things like cytokines, and cytokines would be like interferon and interleukin, and these chemicals, their major use in the body is to actually start the process of the more specific immune response. And so they're going to be very helpful in calling up the helper T cells. So then the next part is induction of the inflammatory response. And inflammatory response includes redness, heat, edema or swelling, and then pain. And then you have the outcome of this would be pus. Now, pus is not actually part of the inflammatory response, but an outcome of it. So let's talk just a little bit about the inflammatory response. So let's say that you get a splinter in your hand, and now that splinter is a bad guy, and it's going to be detected by the different chemicals in the body. It's going to give off signals. It's going to injure the tissues, and the big deal is that mast cells are going to enter that area. And then mast cells, we'll get into this a little bit more later, but mast cells do what we call degranulate. Now, when they degranulate, they release chemicals like histamine. Now, the big deal about histamine is histamine can cause blood vessels to basically do two things. One is it causes vasodilation. So the blood vessel is going to get really large in one particular area, and now because it's so large, everything flowing through that area, it's going to slow down. The other thing that histamine is going to do for us is it's going to create fenestrations. Now, I don't know if you remember from physiology, but we talked about fenestrations before, and fenestrations are basically little tiny holes in the blood vessel, the capillary wall. Now, because of these fenestrations, it allows phagocytes to leave the blood vessels and then to migrate into the region where, let's say, that splinter is and any bacteria that might have got in there, and then the phagocytes, through phagocytosis, are able to eat up and destroy that cellular debris. Now, what phagocytes also do is they actually release what's called HCl O. And that is hypochlorite. And hypochlorite, if you look on your bleach bottle, it'll say sodium hypochlorite, but hypochlorite is basically bleach. And because it's releasing this bleach and it's eating up the pathogens and it's trying to destroy those pathogens, this is how the pus actually occurs. Now, adaptive immunity is when we're able to recognize and react to a large number of microbes, and then, of course, any kind of non-microbial substances as well, like toxins, that kind of thing. And then to distinguish among different types of microbes. And then the other really big thing is it can then remember that pathogen, and once it sees that pathogen, it can very quickly get rid of that pathogen. So with adaptive immunity, when we first get sick with something, our immune system doesn't necessarily recognize it right away. So let's say you get a new virus, the immune system might be a little bit slow to react, which allows us to become more sick from that. But once the immune system knows this virus, it will remember it and really react very, very quickly to it and be able to kill it off, which, by the way, is how vaccines work. So you get a little bit of the virus, the immune system creates what we call memory cells, and then when you actually do get that virus again, it's not like you have some force field around you and you're not able to get that virus. It's just that your immune system reacts very quickly. And so adaptive immunity uses lymphocytes, and then it also is able to identify what we call antigens, and we'll talk about that as well. Okay, so one of the types of adaptive immunity is the humoral immunity, or what we also talked about, antibody-mediated immunity. And this is where our B cells are secreting antibodies. So we use antibodies for any kind of bad guy that is floating around in the bloodstream. So for instance, if you have a bacteria that's migrating from place to place, or a parasite, you want the antibodies to help to get rid of that, okay? In the other case is the cell-mediated immunity, and this is where we're going to be using T lymphocytes. Now, this is typically going to be used against viruses that enter into the cell. So this isn't something moving through the bloodstream. This is actually a virus that's trying to replicate within the cell itself. Now, we're going to use that major histocompatibility complex to help us with that viral replication when it goes inside the cell. So your MHC is a group of proteins that are found on the outside of our cell or on the cell surface. And these proteins can be recognized by our lymphocytes, like cytotoxic T cells. And so what will happen is, let's say we get some kind of virus that gets into the cell, and this virus is going to take over our DNA, and it's going to start replicating its own little baby viruses, you might say. And the cell goes, oh, holy crap, this is bad, we're going to get really sick. And so the cell very smartly actually makes some protein that migrates to the surface of the cell and attaches to these cell surface proteins, or this MHC. That protein that attaches is what we refer to as antigen presentation. That protein is like putting a little flag on the outside of your cell to say, hey, we've been invaded, lymphocytes, you need to come over here and destroy this cell, because we don't want these viruses to be able to get into the system. So the MHC also allows our immune system to recognize self from non-self, or good healthy cells from invaded cells. So MHC molecules are also referred to in humans as HLA. So HLA is the human leukocyte antigen. And there is a class 1, a class 2, class 3, class 4, class 5. We're not really concerned about that. Really what we're mostly concerned about is class 1 and class 2. And these are the ones that are mainly involved in our immune system. So you'll see HLA-A, B, and C, and these are actually categories of genes. So for instance, with human leukocyte antigen A, there are thousands of different types of genes for A. So you'll have A1, A2, A25, and so on. The same thing with B and C. And these guys are helping our cells to tell self from non-self. Class 2, they call them DR, DP, DQ, and they help to do the same thing. Now, these proteins on the outside of our cell are very uniquely ours, by the way, and you have tens of thousands of different shapes of these proteins on the outside of your cell that allows your immune system to know that these particular cells belong to you. So class 1 MHC is found on pretty much all nucleated cells. Class 2, a little bit more specific. These are restricted to immune cells such as antigen presenting cells, B cells, macrophage type of thing. So, and they're found like on lymphocytes. So the class 1 MHC are on the outside of the cells, and they're presenting these antigens to the CD8 cells, which are also referred to as the cytotoxic T cells. The class 2, their major job is to present to the CD4 cells, which are your helper T cells. So macrophage and dendritic cells process and present antigens to the CD4 or the helper T cells, and then they enable these T cells to recognize these antigens, okay? And then this is the initiation of the adapted immunity. So that means that the helper T cells, their big job is to release cytokines that then allow the cytotoxic T cells and the B cells to do their job. All right, so immunodeficiency states. These are going to be problems that people have. The immune system is not functioning appropriately. All right, so classifications of these immunodeficiency states. We have primary and then we have secondary. Now, primary is going to be congenital or inherited. So congenital. That means that I was born with it, but it's not necessarily in my DNA. It's not necessarily genetic. It's just lucky me, whoo-hoo, I have it. I didn't inherit this from my parents. I can't pass this down to my children. Whereas inherited does mean exactly that. It's in my DNA. I got it from one or both of my parents, and there's a possibility that I can pass it on to my children later in life. Whereas secondary means that I acquired this later in life. So for instance, maybe malnutrition or infection, I might have some kind of cancer, or maybe I've taken immunosuppressant drugs like cortisol, other corticosteroids, and those are all going to create this immunodeficiency state. Now, by the way, when we talk about other diseases, primary and secondary still hold for different things besides just immune states. So primary is always going to be congenital or inherited. Secondary is always going to be acquired later in life. Okay, so alterations in the immune system can be immunodeficiency states, which we'll talk about. Allergies or hypersensitivity reactions, transplantation rejection, and then other autoimmune disorders can occur. So, first type of autoimmune deficiency state is a deficiency in B cells, okay, in certain or all antibodies. So B cells are functioning by producing antibodies, which we also call immunoglobulins, and in this humoral immunity defect, it is going to increase your risk of bacterial infection, okay? And especially pyogenic infections, which means these are bacterial infections that produce pus. So humoral immunity usually is not as important as defending against things like intracellular bacteria or fungi or protozoas. Those are going to be your cytotoxic T cells that are going to help us with those. Viruses usually handled normally except for what's called enteroviruses, which are viruses that you have in your GI system. All right, so genetic disorders of the B lymphocytes, approximately 70% of these primary immunodeficiencies are from B cell problems, okay? Immunoglobulin production depends on differentiation of stem cells. So you have these stem cells that have to change into mature lymphocytes, okay? And then those mature lymphocytes are going to generate our plasma cells, and these plasma cells then generate our antibodies. So X-linked agammaglobulinemia. All right, so the first one is called X-linked agammaglobulinemia. Now, X-linked means that this is found on the X chromosome. The A means that it is not there, and emia is in the blood. So this is a recessive trait that affects only boys. There are no immunoglobulins at all being produced, okay? So obviously, if you don't have antibodies, you're going to be very susceptible to many different types of diseases. And the genes necessary for B cell production, okay, is not happening. So you can't convert those stem cells into B cells. And so the child becomes becomes symptomatic at around 6 to 9 months of age. And these symptoms would include things like chronic diarrhea, conjunctivitis, chronic otitis media, which is infection in the middle ear. And then you have pneumonia, bronchitis, upper respiratory infections. These are frequent symptoms that or problems that happen over and over again. Another one is selective immunoglobulin A deficiency. This is the most common type of antibody deficiency. By the way, it's pretty common in people. 1 in 400 people have this problem. They have a reduction in IgA levels, which IgA levels are found mostly in mucus, tears, saliva. Both men and women can have this problem. It's less harmful than other immunodeficiency diseases. 2/3 of people have absolutely no symptoms at all. But if you do have symptoms, you're still looking at types of symptoms like we saw in the other one: bronchitis, diarrhea, pneumonia, conjunctivitis, otitis media, that type of thing. There's no treatment for this. All right, so one type of immunodeficiency is what's referred to as DiGeorge Syndrome, which is a deletion of the gene at 22q11.2. Now, if you look at a chromosome, you have a short arm and you have a longer arm of this chromosome. The shorter arm of the chromosome is referred to as the P arm, and P stands for petite. The longer arm of the chromosome is the Q arm. Then you have in between the Q and the P is what's called a centromere. So if you were to start at that centromere and count downward, okay, you would find this gene. So the 22 on this is the 22nd chromosome. And then counting down from the centromere on the Q arm, if you count down 11.2, this is going to be the gene that you find for DiGeorge Syndrome. Now, this is a partial or complete failure to develop the thymus and the parathyroid gland. Now, the thymus is important for development of your B cells and your T cells. The parathyroid gland is important for production of parathyroid hormone, and parathyroid hormone is necessary to control calcium metabolism. So many different problems can occur, especially due to the lack of T cell production. And you have some changes in the outward appearance of the child too. They have low-set ears, they have what's called a hypoplastic mandible, their upper lip is very bowing, and then they also have a cleft palate. Now, when we're developing in vitro, your palate underneath, your palatine bone and your maxilla are actually separate, and then they have to develop and come together. But these children are born where their palate actually doesn't fuse and come together. So some of the signs and symptoms for DiGeorge syndrome is they can have bluish skin because they have poor circulation, they're weak, they tire easily, they have failure to thrive. So a lot of these children don't survive, failure in weight gain, poor muscle tone, shortness of breath because of that problem. They have delayed development, so they don't roll over, sit up, walk at the same time, delayed speech, learning delays or difficulties as well, and then we talked about the cleft palate. And then they have certain facial features also. So you can see they have the little bowed lips, and then they have more wide set eyes, and so this is very typical of DiGeorge syndrome. Combined T cell and B cell immunodeficiencies. One is called SCID, and this is severe combined immunodeficiency. This is also X-linked, and it is an absence of all T cells as well as all B cells. So basically, they have no real immune system. And this disease resembles AIDS. So this is, again, with AIDS, the immune system has basically plummeted. And with these kids, they just don't have one. So I don't know if you ever watched the movie. It's called The Bubble Boy, and it was a young boy called David Vetter. And David Vetter lived in a bubble that was developed by NASA up until the age of 12. And he never was allowed to leave this bubble. I mean, if he did, NASA actually had a suit for him that he would wear and walk around. And when he actually reached the age of 12, he told his parents that the one thing he wanted to do in life was touch grass. And so they tried to figure out how could they help him out. And so eventually what happened is they gave him a bone marrow transplant. And so the hope was that if they could irradiate his bone marrow and then give him clean bone marrow from someone else who had no disease in their bone marrow, then he would be able to at least produce B cells, and it would give him somewhat of an immune system. So they checked his sister, and it looked like her bone marrow was basically disease-free, so they gave him this bone marrow transplant. But it turns out that she had Epstein-Barr virus, and it was dormant in her bone marrow. And when he got her bone marrow, since he already didn't have an immune system, that Epstein-Barr virus went into what we call Burkitt's lymphoma, and he died within 2 weeks. But he told his parents not to be too upset because he was very happy that he was able to play and run in the grass for a little while. Another one is Wiskott-Aldrich Syndrome, and this is a rare X-linked disorder. Both the B and T cell immunity is impaired. They have a complete failure to produce antibodies, but that's not all antibodies. It's just a certain class of antibodies, and they get recurrent infections. They get hemorrhage because they can't produce platelets very well, so they have what's called thrombocytopenia. They have eczema, and then they have petechiae. Now, petechiae is where little tiny capillaries underneath the skin start to burst. They have otitis media quite often as well. So you can see the picture of the petechiae, and they're little teeny tiny dots underneath the skin. Whereas if you get groups of these together, this is what we call purpura. Now, you've probably seen purpura even more when you look at like your geriatric patients, and those patients have big black bruising looking things under the skin, and that is actually purpura, where the capillaries are breaking down and bleeding, and it's not necessarily that anybody has done anything to them. So some of the clinical features are they get opportunistic infections, which means, for instance, pneumocystic carnii is in our lungs already. Candida albicans is on the surface of our skin, in our mouth, and because we don't have the immune system to keep these down, the infections take over. Some people will even have herpes simplex or varicella virus, and they can die from this as well. Bone transplants, again, like taking the bone marrow, transplanting that into the patient, could be effective to help them to produce more B cells. Now, the complement system is a system where the liver makes 9 different types of proteins. And these proteins can then help us in a couple of different ways. And one of the ways is that these proteins can actually form rings in the cell membrane. And those rings kind of look like a little donut. And what happens is it opens up the cell membrane and allows extracellular fluid to go rushing into the cell and then causes the cell to burst. And we're really going to use this complement system when the cells have been infected by viruses. Another thing that this complement system can do is that opsonization that we have kind of talked about before, where some of those proteins attach to certain antigens on the outside of the cell and then allow phagocytes to bind to the cell and to destroy the cell. And then also the complement system can attract those phagocytes to the cell, so that's chemotaxis. And then the extracellular fluid rushing in causes the cell to rupture, and we refer to that as lysis. Now, sometimes also it causes clumping of antigens, and so that helps to prevent things from spreading through the system. Or we'll talk about it in a little while, it may create problems as well. So disorders of the complement system, first we have primary, and this is mostly transmitted as autosomal recessive traits and can involve one or more of those complement components. And then secondary can occur in persons with functionally normal complement, but it can be caused by other problems like diabetes, leukemia, immunosuppressant drugs, especially things that may affect the liver as well, because the liver is important in producing these complement proteins. So one of the proteins, which is C1, complement 1, has inhibition, and this is an autosomal dominant disease, and we call this hereditary angioedema. And you can see that what happens to these people is they have a lot of swelling when sometimes just the littlest things occur. So we're going to watch a little video on this.
My name is Christy Harden. I live in a small town in southern Indiana called Corydon with my husband and my daughter on a small farm. I've been a nurse for about 12 years, and I'm currently back in school to finish my bachelor's of nursing. We have about 25 members of my family living with HAE, and it's affected 5 generations of my family. My daughter's the 5th generation, and many of that 25 all live close to us in this small town. Hereditary angioedema, or HAE as it's commonly referred to, is a disorder where you don't have enough of a protein or your protein doesn't work properly, and it can cause asphyxiation. When I had my first attack at 13, it was a facial attack. I looked into the bathroom mirror and my face was swollen so badly I didn't even recognize my reflection. And I went and told my mom, I'm like, look at my face, what's wrong with my face? And she knew right away that it was HAE.
It was called a swelling disease in our family. Of course, they told us not to have kids, and my daughter and my boys seemed to really suffer with it. It seemed to affect their lives. It makes you feel kind of guilty. And then when my grandbaby has it, Cammie, I hate it for her.
The most difficult thing growing up with HAE is all the stuff you miss out on. Like, my friends would invite me to go hang out, and then I would get too excited and get sick and have to cancel.
No one outside of my family understood what it was like to deal with HAE and the attacks. It does make you feel lonely when you get left out of things, and it makes you feel frustrated that, this is a difficult disease to understand.
My friends really do try their best to understand, but I don't think they really can, because I can tell them and they can try to compare it to when they've been sick, but it's just not the same.
Being a patient and also a caregiver for someone with HAE is very difficult. And even on days when I feel perfectly healthy, if my daughter has an attack, then that stresses me out because I'm so concerned about her, then I end up getting an attack. So a lot of times both of us depend on my husband.
My dad is like super dad. I don't know what I would do without my dad. And my mom, also super mom. She's always supporting me and ooh.
As I've gotten to know the employees that work at CSL, they have been so compassionate and patient focused. That's why I made the decision to become a patient advocate for them. Some of the most touching and rewarding things that I've got to do as a patient advocate is talking to a patient who told me I've never met or spoke with another person that had HAE before. And that just blew me away because I had such a big family and I had so many resources, to talk with someone with HAE.
I think we have a good support system because we understand. We've all been there.
My grandma is easily my best friend. She really understands that it's hard for me, but she also doesn't let me make that an excuse to not do something.
I love her dearly. She's one of the highlights of my life.
Although I had a much different childhood than my mom, Cambridge has a much different childhood than me. We all have a bright future because of the advancements that are being made in the future of HAE.
So a phagocytic system, the definition is composed primarily of what are called polymorphonuclear leukocytes, or PML, and then mononuclear phagocytes, which are basically what you would think of as phagocytes. So these cells migrate to the site of infection, they aggregate around the affected tissue, and then by phagocytosis they envelop it, and then they're able to destroy it. And I thought maybe you'd like to watch this because this is kind of cute. So this is a phagocyte following a bacteria moving around in the system. So dysfunction of the phagocytic system is going to be usually a reduction in the number of phagocytes that are available for the system, and this is something that makes you more susceptible to things like candida, okay, or certain fungi. And then you have chronic granulomatous disease, and this then becomes things like impetigo or recurrent pneumonia. You may also see skin abscesses and rectal abscesses, and so this is impetigo. Not a fun thing to get. And so that's actually a YouTube video that you can watch on stem cells. We're not going to watch this right now. But basically what happens is you need these stem cells, which are cells we call undifferentiated cells, and these stem cells are then going to become the types of cells that we need in order to fight off disease. And we can take some of these stem cells and transplant them into donors who have this disease, like SCID or Wiskott-Aldrich Syndrome. And the hope is that once it establishes itself in the bone marrow, they will then become cells that are able to produce B cells and antibodies for that person and prevent the disease from happening. All right, so we are going to talk about hypersensitivity reactions in the next video.